Good afternoon, everyone. Welcome back. This is session 5 on population genomics and phylogenomics. So there are two talks in this session. The first one is by Professor Binay Panda from JNU. He's going to talk to us about X-plat, a cross-platform transformation tool based on the nearest neighbor joining framework.
Thank you, Tej, and thank you, Chirag, Devnath, Sanchit, and the organizers for inviting me. And I think it's been a good 2 days. So what I thought I'm going to do in the next half hour or so is to take you back, and that will tell you that I am old, take you back to the era when there was no sequencing. How did you do genomics? And in the era when there is sequencing, what do we need to do to use the legacy data? In fact, this is a disclaimer. I just realized that although I'm going to show you about a tool where we use the nearest neighbor joining method, which mostly people use in phylogeny, but my talk has nothing to do with phylogeny or population genomics. But with that, if I can have the, okay. So we work primarily on solid tumors, particularly head and neck tumors. And some of you might know India has a particular problem with head and neck cancer because of extremely high incidence and mortality. But today I'm going to talk about a tool, less about the cancer. And the second part of the lab actually works mostly on metabolic engineering, so I'm not going to talk about that. So the tool that I'm going to talk about is primarily used how multiple people in the genomics community can go back in time and use legacy data. Because there are a lot of legacy data. I mean, yesterday you heard, I think Saket was talking about single cell genomics. The field is moving in a pace is sometimes very difficult to keep up with. However, and this is very important, the utility of the tools in the clinic has still lagged behind decades. So people are still not using qPCR in the clinic, but we have moved to single cell genomics in the research. So there is a huge lag. And because of the lag, there's a huge amount of data which is already generated, deposited in the database, not useful anymore. So I'm going to talk about how to use the legacy data, make it comparable so that you can use it for validation. Okay, so I'll start with the problem. Where exactly is the problem, and a little bit about the background and what we have done and why we think this tool is extremely useful, very simple, but extremely useful tool for biologists and the clinicians. And then where does the future hold? So before I start, I must acknowledge the people who have done the work, who enabled us to do the work, and where the funding came from. As the name suggests, the work is primarily done by the postdoc in the lab, Nirja, on the left, and a student, Sarah, on the right. So primarily is their work. I'm just the talker here. And the enablers are the star work. I'm an extremely big fan of what Lucas did in 30 years back, which is unimaginable. But you could see that everything is coming to fruition now. So he was a thinker, he was an enabler, he was a dreamer. And of course, the funding agency, this was funded by DBT. And the human samples that I'm going to talk about, the breast cancer samples, was done in collaboration with Lars's lab. Lars used to be in Technical University in Denmark, where I spent a few months, but he has recently moved to the Copenhagen University in Denmark. And the idea of working on a tool that will actually allow to use the legacy data came when I was teaching a course in UC Santa Cruz long time back. As you can see, it's like 35 years back in molecular diagnostics. And then I realized while that time, the microarray was just coming up. In fact, I was very fortunate to work with Steve Forder, who was the inventor of Affymetrix, and I'll talk a little bit about that how people have moved on, but the tools haven't moved on, but the clinical community hasn't moved on, and hence it never really helped the diagnostic community. So the idea of using a tool so we can convert back and forth actually originated during those times. Okay. This particular work that I'm going to present today was presented as a poster in RECOM 2023, but the good news stopped there because right after we presented the poster, we came back, we wanted to finish the validation and send the paper. The NSM server, I was telling Devnath yesterday, the server conked off. So we lost everything, particularly the data. Code, of course, we had. But unfortunately, GDC, which is the database for all the human samples, stopped giving access to the raw data right after that. So we had no idea that we not only have lost the data, but we had lost the ability to download the data. So the cycle starts again. So we had lost 2 years of complete work. Actually, the grant was for 3 years, so 2 years of data and the work is lost. But this is life. So anyway, so I'm going to tell you about why we could not actually replicate some of the findings that we had in some of the species and to have to redo the entire work once again. Okay. I'll take you back in history, and particularly for the community who are not biologists. It's a very important lesson to learn. Many times we work in silos, and this is one of the problems that we particularly in India have. The hardcore CS people don't talk to the biologist, don't understand the biologist, and vice versa. So we are stuck in our own silos. Now the problem with working in silos is that the tools that the CS community or the algos that the CS community comes up with are very good. The assays that the biologist community come out is very good, but there is no intersection. And therefore the clinical and the translational utility of those tools is text to zero. And this is exactly what we are going to do. We means the community as a whole. We need to. I'm very happy to tell you that I have seen a change in the last 10 decades. We are moving closer. We are not there yet, but I think we are moving closer. So I think in the future, the idea is to not work in the silos, but work in team. And I think yesterday I was telling someone that it will be really great to think about a problem. Like say, we have a diagnostic problem. How do you diagnose a particular microbe in the clinic? Can we actually assemble a team of CS people, biologists, genomics, bioinformatician within a problem-centric team rather than a tool-centric team? Because right now what is happening is that we work in a tool-centric team. So we never talk to each other. And I think I'm happy that we're moving in that direction. Maybe in a few years we will be there, but that's basically the aim. So many of you may know the field started with qPCR. And I will define what qPCR for a laymanish term. I'll take you back to the history. I'll give you a lot of history lessons here because I really love history and I wish I had a good history teacher in high school, which was lousy, and otherwise I would have actually been a historian. History teaches us quite a lot of beautiful things, okay? And this is the reason I'm a true believer of the social science and humanities existing with sciences. We learn so much from each other. Now, many of you know probably already that Gary Mullis, the guy who got Nobel Prize for the invention of PCR. PCR is nothing but you have a piece of DNA, a short stretch of DNA. You design short oligonucleotides for both ends, and then you amplify. Now, this amplification has to be anchored by an enzyme called DNA polymerase, which can withstand a change of temperature of melting. So you melt DNA at high temperature, close to 95 degrees, and then you got it back to a temperature where both the strands can anneal together. That's annealing temperature. So you go back and forth. You melt, anneal, amplify, melt, anneal, amplify. And this is a Nobel Prize discovery when he was actually driving from Redwood City with his girlfriend, then Matt Fiona. Now, since then, we have moved a lot further of using PCR in clinical diagnostics. Now, I'm talking about qPCR. The difference between PCR and a qPCR is that Q part, which is the quantitative part. That means in each cycle, I can actually quantitate how many copies of DNA or RNA converted into DNA has been incorporated in my reaction. So this will give you, many of you, it's probably become a household name right now, particularly post-COVID. You know, all of you got tested by qPCR. qPCR was nothing, a method where you isolate RNA, convert that into cDNA, which is a complementary DNA, and then you put that piece of cDNA, or X copies of cDNA into tube, add your enzymes, add your substrate, add your cofactors, and try to amplify it. As you amplify in each cycle, remember I told you up and down, melting and annealing, each cycle gives you a quantitative output of numbers. And that relates to how many copies of viral RNA was present in the beginning, and hence you are called positive or negative. This was the beginning of gene expression era. This goes back, of course, in late 80s and early 90s. Then came arrays, microarrays. This was a big splashing news, and I'll tell you a little bit about history. Now, then came sequencing. Now here, and I'll give you the array and sequencing background as I go forward. The size of the letter represents the amount of data present in those times. If you are in 1990, the amount of data which was generated with qPCR was largest because array was just beginning to peek his head. As you go in 2000, arrays dominated the field. Everything was generated by arrays. As you move to 2010 onwards, sequencing took the larger share. So as you can see, as you moved in the last 35 years, the data which was generated in different platforms, the amount of data that generated different platforms varied. However, these guys became obsolete, and so did arrays. Database is filled with that. We can't use that. So that's the whole idea. If I am right here in sequencing, can I actually come back to use the data here? It'll be really nice. And that's the whole idea. Now, how do we do that? Okay, this is the same graph represented in circle. The size of the circle represents the amount of data proportionally, not exactly, and the timeline, as you can see on the bottom. So how do I now go back and use this different data set at the same time? If I'm using clinical samples today to understand cancer, understand AMR, antimicrobial resistance, to understand anything else, I have limited number of samples. But I have a huge amount of samples in the database that people have typed. I can't use that. Can I actually use a tool where can data A can be converted to data B or data C? Now, today I'm not going to talk about data A. The tool primarily is going to talk about data B and C. The reason being is data A is a very low throughput data. You only use one gene at a time. The high throughput data is primarily an array and sequencing. You know, Sidney Brenner used to say, he's obviously a very famous biologist, and I think Miley is not there. He would have told, he used to joke about high throughput biology. He used to say he never liked high throughput biology, and he used to say high throughput biology is just become high throughput but no biology. And the idea is that finally anything that we do has to be useful biologically or clinically. Okay, now I'll take you back to the array history. The microarray was invented in 90s by two independent group of people, both in the Bay Areas, one by Pat Brown's group in Stanford. And in fact, how many of you have heard about a company called Impossible Burger? Okay, that's the guy. Can you imagine a biochemistry tenured professor who invented microarrays have left, now has actually started an Impossible Burger? That's him, okay? But he came up with a system which is very easy. This was, you remember 1990 was just the time when human genome sequencing was getting done. Before that, only a few microbes, model organism sequences were known. The idea is to take total RNA from a sequence organism, convert the RNA into cDNA of varying length. Some genes are X kb, some genes are 10 kb, some genes are 1 kb. And then you spot those on a glass slide. In this particular case, you take a test sample. Let's say a test sample is your cancer sample. Control is a normal sample. Once you do isolation of mRNA, you label those to two different dyes, in this case Psi3 and Psi5. You can label them anything you want as long as they're different. Now, if I mix those together and hybridize on a glass slide, and this glass slide has the entire complementary cDNA of that organism, the expressed cDNA. Now, based on the complementarity, wherever I see a signal, I know that particular RNA was expressed. Now, depending on the color, either it's going to be blue or red or mixed. And that mixed intensity can be quantified. So this, for the first time, gave an idea that not only we can understand expression of the entire set of genes in an organism, but you can do that at one time. So this was a significant milestone in the study of gene expression. Because remember, before that we used to do qPCR, one gene at a time, maybe multiplex, few genes at a time. This gave us an idea about studying gene expression at a genome-wide level. Now, at the same time, just 25 miles across Palo Alto was Steve Forder. And I'm very fortunate I was a part of Steve Forder's initial group when this started. Now, Steve had a revolutionary idea. At the same time, very simple idea. So what Steve did, he leveraged the semiconductor technology. If you remember, I mean, Sanchit would know the office where Intel is, the series of high-tech offices in Santa Clara. And Affymetrix was right next there. So being a biotech company among the Silicon Valley, so-called the high-tech companies, was a little bit odd. But at the same time, he utilized the power of semiconductor making in biology. So in simple terms, the way wafers were designed to make computer chip, he used the same wafer to design a DNA chip. And in fact, colloquially, these were called as DNA chip. This was nothing but just imagine a 1 inch by 1 inch wafer. This is like a computer chip. This is just a casket around it, where, like what Pat Brown did, deposited shorter oligos. Instead of having a long cDNAs, deposited 25 mer oligos. And those 25 mer oligos represented each and every gene in a genome. That means that one of the prerequisites for this technology was understanding of the genome. Sequence was known, right? And instead of what Pat did, isolating normal and the disease samples and labeling them 2 different colors, here you just use 2 different arrays and then compare their signal. So then fast forward to, so we are still in 1990. The mid-1990s, Mark Chi, and I'm giving you a lot of history. This is only the guys who are actually neck deep in genomics would know. He used to be a postdoc with Steve. And then he's an Australian guy. He was in the Bay Area. He went to San Diego to start a company called Illumina. Illumina actually was a very bad company then. It's not unbelievable by many now. Illumina didn't start with sequencing. Illumina acquired a technology to start sequencing. Technology was made by a company called Solexa. It's a UK company. Anyway, Illumina's first bread and butter was actually SNP genotyping. They were not in expression. But the way it worked is they used a bead, which has an addressable address. This is again an oligonucleotide, which is fixed, same, and then you have a probe. This probe sequence varied, but it's a short oligomer, and each probe corresponded with every single gene in human genome or any other genome that you want to interrogate. You have the labeled cRNA. You remember how Pat used to make cRNA, and even the Affymetrix used cRNA. It's the same cRNA you hybridize, and based on your address, you would find out, deconvolute the message to find out the SNP or the expression of the gene of interest. Now, this is how it worked in the spotting array, which is basically the Pat Brown's array, as I told you. You have probes, you have targets. Targets are labeled. Targets are like human RNA converted into cDNA, labeled. If there is a complementarity, you have the signal coming out from that, and you know the location of each probe. You remember that 1 inch by 1 inch slide that I showed you? It's just a matrix. You know the location of each of these thousands of probes, and based on that location where the signal comes from, you deconvolute to find out whether the gene is expressed or not, and if it is expressed, how much it is expressed. This is how the Affymetrix is going into detail. Instead of having a single probe, the technology devised around 2 probes with a single mismatch in the middle. The probes are 25 mers, so the exact center is number 13, position number 13. So you expect in a best case scenario, you will have a signal on the perfect match, PM, and you will have no signal on the mismatch. So a PM divided by MM for a series of probes here will give you the signal for a gene of interest. Okay? Now this is how the sequencing assay works. You are very familiar with this. You have been hearing for the last 2 days. You take the total RNA. This is again the whole transcriptome RNA-seq, because there are so many RNA-seq. This is the whole transcriptome RNA-seq. You take the total RNA. Ideally, you use poly-A RNA, but you don't have to isolate poly-A RNA. And then you fragment, you ligate an adapter, which is a short oligonucleotide sequence, and then you use that for your sequencing library preparation. And then this is essentially what the readout you get. Based on the depth, you have on one axis that is expression level, you can count, and on the y-axis is your genome coordinate, chromosome at a time. Okay. Now coming back to what we have done, I've hopefully given you a little bit of background of how this technology worked. And the point I'm trying to make is the hardware, the software, and the way the targets were made for all this technology was very different. And that is the reason why people could not come back and use a technology to interconvert. So what we did is, I'm not going to go into the detail of each of the method. The reason I'm not going to go into detail is that it's very optimized method. We have taken the methods which are off the shelf. We haven't invented new this method. We have optimized some of this method. But these are very standard methods. Anyone who has worked on arrays, anyone who has worked on expression or methylation. So what I'm showing you is only for expression and methylation. That means if you have the whole genome expression data or you have whole genome methylation data, you should be able to convert back and forth. 5 minutes, okay, I'll try to finish soon. So the way it works is actually simpler than it sounds. What we do is we take samples in one axis, genes in another axis. You see G as genes, S as in samples. We take for array and we take for sequencing, okay? Now, the prerequisite of building this tool, which was very difficult, and I'll tell you during the panel discussion why the data is very critical. You need paired data. What is paired data? That means the same data which has been used for array sample, same sample has been used to generate array data, and exactly identical sample has been used to do sequencing data. So sample remains constant, it's a prerequisite. In that case, then what we do is that we do a split, in this case 70-30 split, where we have array data in one axis, sequencing data in another axis, and we are trying to join the neighbor, nearest neighbor, as people do in phylogeny. And in the next step, what we do here is very, very simple. We estimate the slope and the intercept for each point. Once we do that, we actually calculate the root mean square error. Once we calculate the root mean square error, we do that 10 random times and take an average of the 10 random times. And therefore, we get a value of sequencing from array. You reverse that, you get from array from sequencing. Now, it's simple, but it works. Okay. This is the data for the ground truth. All of this data, as I told you, the identical sample has been used to generate array and sequencing. So we can compare to one another. So I'm not going to read the detail. As you can see, the exact array platform and the sequencing platform that we were used to generate the data. And we also used for validation the breast cancer data because we wanted to use the clinical data. This is the data which came from Lars's lab when I spent a few months there. This is a breast cancer set of 62 samples. Again, there are multiple different subtypes of this breast cancer data. And any of you who know breast cancer knows that breast cancer subtyping is done based on the hormonal status, gene expression of certain particular interest genes. And we use these 2 samples also to validate the tool, that can we do interconversion. Okay, how does it look? Very quickly, because I have very little time. On the left-hand side, I'm showing you a comparison of array to sequencing RMSE. In other words, lower RMSE is good news. That means low variation. As you can see here, there is very little variation. Don't go by the spread. Of course, there has to be a spread because it's technology 1 and technology 2. But the majority of data is actually in the low RMSE range. On your right-hand side, I'm showing the same thing. Here, this is array with the number of genes. But on the bottom, on the color-coded, I'm showing the sequencing pattern. So as you can see, most of our genes are in the first 2 divisions of the data. Means, again, low RMSE. Same thing for methylation. If I compare the array to sequencing RMSE data, it also comes in the lower quartile here, very small variation. Similarly, most of the data also belongs to low RMSE for sequencing if I compare that to the array. Okay, how does it look if I compare to expression range? Expression range is simply because it's defined as a max and min. Now, as you can see, that the measure of variability is also low. You would expect variation because the way, if you remember, I described the way the array defines expression and the way the array defines methylation is very different. In case of an RNA-seq or the whole genome bisulfite sequencing, you have reads all across the gene. In case of array, you don't. You have only few probes, and that too, which is biased towards the 5' end. But despite that, you have remarkable consistency of less variability if you compare to array range, sequencing to sequencing range, which again is actually very good news. And then we wanted to compare, does the actual value of expression correlate with the transcript length? And the reason we wanted to correlate transcript length is because in case of a microarray, the length of the transcript is a very important parameter. Also for sequencing, as you know, we do, obviously a lot of people now use TPM, which essentially cancels out the length of the transcript, and that's essentially the purpose. That means even if you have a long gene or a short gene, you can rely the data. You can rely on the data because the length of the gene has nothing to do with the actual quality of the data that you get from expression. So same here for both for sequencing and array. And finally, we compared the RMSE from both array and sequencing with the length of the transcript. And here I'm showing you overlapping plot. The darker plots are the one which has basically the longer transcripts, and if you go lighter, which are the shorter transcript. And in fact, it holds very good even for very long transcripts, the very low range in RMSE, which again is a good measure for interconversion. Okay. Like any tool does, we have to do a comparison because the reviewer is going to ask. Now, to me, this comparison is meaningless, but we still have to do it. Now, okay, no time. I'll finish very soon. Just give me a couple of minutes. We tried to, Casey Green, many of you in the community might know, he's moved to Boulder now, University of Colorado, I think, a different campus. He devised a tool called TPM, which also is used for interconversion. So we took his tool, and the other tools we couldn't take for a variety of reasons. I'll tell you that both, if you compare our X-plat for array and for sequencing, and his TDM for array and sequencing, obviously you can see that ours perform better. And this is probably the last slide. I have a comparison. Okay, this is the reason it does better, because in their case, there's a huge spread of the RMSE, and these are the guys who actually skews their results. And in our case, it's not the case. Okay, this is the final slide of comparison. I'm not going to read it, but I did not show you any results with these three comparison tools because these tools are extremely old. They do not work for methylation, was not designed for methylation, and also it's not incompatible. Many of the cases where the grad students are moved forward, the postdoc, the tool is not available. So we just used it, but then because we lost everything in NSM server, that comparison is also gone. Now we have to see if we can somehow get some of the data to actually reuse. But the bottom line is, you just have to see we can use both expression and methylation, plus we can interconvert array to sequencing, sequencing to array, which except one tool others don't do, or only do expression. So I think we have an advantage over others. Now, with that, if I may take just 10 seconds, this is the old comparison that the data is lost, so I didn't show that in the beginning anyway. Okay, in conclusion, we have a tool, simple, works. It does conversion in both ways. Sequencing to array, array to sequencing gives us an ability to use the legacy data. Okay, just 10 seconds, nothing to do with our work. This came out 2 days back in Nature. Okay, I don't know how many of you have seen this. This is about how we practice our science. It's essentially, as the topic says, you rise being a conformist. So many of your students, if you disagree with your boss, you are screwed. If you disagree with the big guys, you're gone. That is the only way to rise in science. And there's a Nature paper on that. It's not I am saying. There is a huge pivot penalty. Pivot penalty basically means how much of deviation you are doing from the work that you have been trained on. That means after 10 years of grad school and postdoc, if you want to start in a new field, you have a distinct disadvantage. Because people say, I don't know what he's working on. His background is different. But this is exactly what we're not supposed to do. We're supposed to be heretical. But science doesn't unfortunately move like that. So be heretical. That's first point. The second, practice open science. Obviously, the CS community is very good and the physics community is very good at putting things in preprint. Biology community is lousy. They're extremely cagey. They don't share. share preprints, and I would highly, highly recommend if some of you want to be involved with an organization that I am very active called GF4GH. GF4GH is extremely good in making standards for genomics. Like Genome in a Bottle, they make standards for all genomics community, tools and standards. So if you can be involved, I recommend do get involved, okay? And I think finally just, advertisement. I have a YouTube channel. I try to interview some interesting people. As you can see, the head of EMBO, Fiona Watt, Mike Eisen, the ex-editor-in-chief of eLife, Ivan Oransky, who is the head of Retraction Watch, Phil Altbach is an educationist in Boston, and Sholto David is the guy who actually exposed the corruption in publication in Sloan Kettering Cancer Institute. So I think with that, thank you very much. Love to take any questions.
We have time for a few questions.
Hello, sir. I'm Anjane, and I liked your talk. I had a few technical questions. So you have used RMSE as a measure of error. Did you also try other measures like mean absolute error, or what was the reason behind using RMSE?
Yes, short answer is yes, we did, but RMSE performed much better.
Okay, and in one of your plots where you were showing the expression data, you had the sequencing on the y-axis and the array data on the x-axis. So the RMSE for the sequencing data was much larger as compared to the array data. Usually we would expect that since sequencing is more recent, the RMSE should be lesser since the method would be more accurate. But what could be the reason behind that?
So you are talking about, for example, here.
Yeah, this one, yes.
Yeah. So you were saying why this data? Say it again.
So I'm saying that for sequencing RMSE, we see that the range is like the values are much.
They don't compare the range. They are completely two different things. You should not. In fact, that was one of the first things that I also noticed. The RMSE, the way sequencing technology works is completely different because you don't compare the range here to range here. You can only compare the spread. That means if you have a huge RMSE here, anything which is here is bad, of course. That means which is large. Just don't compare the scale here to the scale here. You can't. One is technology one, the way it discovers the genes and the RMSE calculated from the absolute number. Here is absolute numbers are derived by the reads. So you can't compare. Compare in which quartile your data falls. If it falls here, then obviously it's bad for both. If it falls here, then it's bad for one, not the other. If it falls here, it's bad for the other, but not the one. So this is basically the idea. Idea is to compare the RMSE without looking at the actual scale.
So the way the RMSE is calculated for both the technologies, is it different or is it?
No, RMSE is calculated the same way, but each technology has a completely different number. You can't compare. The sample is the same. That's the whole idea.
Yeah, so my question was, since sequencing came later, my conception was that the sequencing would have lesser errors, so the RMSE orders, like the values, absolute values should have been lesser than the array one. But here we see opposite.
Hold on. You can only say that if you compare for the same gene. You don't know. That gene level identification is missing here. You are right. You are right that you anticipate sequencing to have a lower RMSE.
Yes.
The reason being is that you have many reads.
Yes.
Right?
Yeah.
Yes, that does happen in many cases. If you really see, for example, here displayed, there are more genes here. You do see that, right? But also remember one thing. You are collapsing the data. You are taking multiple reads corresponding to a single gene as a single point. But perhaps that single point related to the sequence, that is the reason why we compared. If you see this particular graph, why did we compare the transcript length? Essentially for that reason. Because you anticipate for a longer transcript to have lesser bias for sequencing than array. But at the same time, we also know that in case of an Affymetrix array, the probes are designed in the last 600 nucleotides. So it doesn't matter if you have a 50 kb transcript or a 5 kb transcript or 1 kb transcript. The probes are in the last 600 nucleotides. The reason it's in the last 600 nucleotide is when polymerase actually starts polymerizing, the enzyme drops off after a kb. So we know even if there is a difference in which these two technology work, the final output is probably more or less same. I would anticipate, which we haven't seen, the difference for spliced transcripts. Because in case of a spliced transcript, I don't anticipate to see better data with RA, but I do anticipate to see much better data with sequencing.
Okay, thank you.
Sir. Sir, thank you for the talk. Sir, I want to understand why should we even spend conversion of biological data in one format into another format in the first place, rather than if I already have the data in one format, why shouldn't I proceed with analyzing in that format?
Why would I consider that? Means you didn't understand anything I talked about. The reason you want to use conversion is, so let me give you a colloquial example to you. Maybe it will make sense. You have a Maruti 800 for the last 25 years. When did Maruti 800 get introduced in India? In 80s, when Sanjay Gandhi did it. Maybe you're too young, you won't remember. So Maruti 800 was a car which was imported initially from Japan. Then it started making, Suzuki came here, Maruti was formed and did. Suppose you have been driving that car. It's an old car, it was not automated, it didn't have power steering, all these things. Now you are driving, I mean, say the most recent, that Mahindra's electric car. It's still a car, right? It still takes you from place A to place B. Now, if I am a Mahindra showroom salesperson, you comes to buy this and you say, yes, I own a Maruti 800. I said, no, that's a piece of junk. It's no use. I will teach you how to drive my new electric car. He said, but I know already driving. He said, no, it doesn't matter. The steering wheel is different, the gear is different, the gear ratio is different, how you use the car is different. You have two mirrors. In Maruti 800, you had only a central rear view mirror. You didn't have any side mirrors. So I'll have to teach you. Okay, how about the guys, rather than saying that, you said, sir, you have used a very old car. Here is a YouTube video for 10 minutes. It will rewire your brain. If you would have used left-hand steering this way, what to do with a new car? Would you not like it? That's the reason. You have a lot of data sitting in the database, totally rendered useless. Now, if you can convert this data, then you can compare. You can use for the large validation set. You can use for testing. But now you cannot. So the whole idea is to use legacy data to improve the sample numbers. Is that clear?
Yes, sir. Thank you.
Thank you.
Hi, sir. Sir, so we had used TDM once just to convert from seq to microarray. But the thing is, like, one thing, when we want to use these data for machine learning purpose and all, we're just concerned about this annotations. That is, we have probe IDs when it comes to the microarray. There might be different technologies there. Then in the sequencing also, there might be different conventions of naming, and also platforms are different. And so
Absolutely.
how is that managed in X-plat?
Very good question. You must be comparing apples to apples in this. That means if you have used one version of the genome, if you have used the T22T for one, then you must compare the T2T for the microarray. So, for example, that is probably the reason why we had to go back to HC19, because most of the arrays were done, but there is no way one cannot actually move back. But then it's a lot of work, we're just lazy. So we thought, let's just do with HG19, which is already done in array. For sequencing, it's just a leftover. Simple.
No, but sometimes what happens is some of the data sets, like, because sometimes we might have data set which is split in both the cases, but that means we will have to use only one set of data.
No. You can lift over. That's what I just said.
Okay.
This is a set of coordinates, right?
Okay.
You just have to make sure that all the data is aligned or annotated against the same version of the genome.
Okay.
Then it's no problem.
Okay.
But you'll have to do that.
Okay. Like in the if there's a GUI for this, so
Yes.
that doesn't automatically do, right?
Yes.
Or that automatically does.
That that doesn't do interconversion. So far we have built for HE19, but there is no way we should also be doing it for 38 and T2T.
Okay.
That's not a big job.
Okay. Thanks.
Yeah.
Okay, let's thank the speaker again.
Thank you.